thank you everyone for the invitation to come. So I'm actually a basic science researcher. Everything I've been hearing about has been very patient-centric. I'm about to blast you guys with a bunch of bench work. So strap in, I'm sorry. But I would like to introduce you to the Broad Institute's Drug Repurposing Hub, and it's a resource that we've put together about 10 years ago. But I don't think a lot of people know that it exists, and so I'm here to rally it a little bit. It's obviously a source that could be used to find new therapeutics, but we also want to talk about the fact that it can be used to highlight insights into biological pathways that might be involved in your disease of interest. So the Drug Repurposing Hub is a collection of small molecules that was built about 10 plus years ago. The reason we started building this is because we realized in this research community there was a lack of a centralized location to get FDA approved FDA and globally approved compounds, clinical candidates, and preclinical tool compounds that could be used to study the idea of repurposing in a cell-based or a biochemical-based model. So, you've heard a lot to this conference about patients researching the literature, looking for drugs that have been repurposed in their disease. Somebody actually has to do that research to get to that publication. So this is where we thought there was an unmet need in the research community. So this was a philanthropically funded effort at first. We had a postdoc that dug through the literature and identified all the compounds that he could to put into this library. You can imagine this is quite a task, somebody having to dig through the literature to understand what's in the FDA, what's in clinical trials, what tool compounds are out there for targets that are not in either of those collections. So this was a massive effort to identify and purchase all of the compounds. Anybody who's ever tried to purchase a compound, that in itself is a feat. And then to go along with the actual procurement of the compounds, each one was comprehensively annotated for their known activities, their mechanism of actions, their indications. And then when we brought those compounds in, we actually experimentally confirmed the purity and the identity of each of those compounds. For many of you who do basic science research, just because you buy a compound from Sigma does not mean that that is the actual compound in the vial. So there was a lot of concerted effort to build a high quality small molecule library that was cohesive, well annotated. And in 2017, that library was published, and at the time it consisted of 4,700 compounds. The idea was to be able to offer this to the academic and nonprofit community so that you could screen this library to find new indications for old drugs, the very definition of repurposing. So as of 2025, we've actually gone through about 3 iterations of this library, each time adding compounds, taking compounds out, really deeply assessing the quality of this library at every turn. As of 2025, last month, the library was just recently replated. And so now we are at about 5,500 compounds. The website is up there if anybody wants more detailed information. The library is broken up into 2 subsets. Our Repo 1 subset is about 2,800 compounds, and these are all our FDA and globally approved compounds, including some withdrawn compounds. And the other half of the library is what we call our Repo 2-3 library. Repo 2 encompasses clinical candidates at the time of annotation, and Repo 3 are preclinical or tool compounds that cover target or MOA space that's not currently covered by the FDA and clinically approved clinical candidates. So you can imagine this is one of our most popular screening libraries. As the head of screening at the Broad, this is the library that most people come to talk to us about, and it's the first library that most people tend to screen in all of their screening campaigns. So the website, if you go to it, you can maneuver through it, you can download information. It will give you the name, the smiles, everything that we know about the compound that we've annotated. You can look at current versions, historical versions to see what's been in there, what's not been in there. You can actually navigate your way through the data portal as well. You can search by compound name, targets, phase, anything like that. Really, it's whatever you want to learn. You can kind of dig through this website. We're in the process of updating this, as you can imagine, given that we have a new version of the library. So give us like another month or so, and then it'll be better updated. And if you actually click on one of the compounds, when we say annotated, we really mean annotated. We had a team work to put as much information with all the sources that we can about this compound. So, we really want you to be able to understand what you're screening. All right, I'm not going to bore you with too many details, but you can imagine quite a few people have used this library for a multitude of reasons. Again, as we said, the primary reason is for people who are looking for new drugs all drugs for new indications. Several of them are here. The library is accessible to all academic and nonprofit institutions. Obviously, it's a disease agnostic, right? You can use it for study any disease, and it gives you not only just therapeutics, but also biological insight. And there's a few examples up here that I won't dig into, but the last one is probably the more interesting one. We had a collaborator screen this in renal cell carcinoma. It's a rare cancer that's induced by TFE3 oncofusions. And he was able to use this library to look for compounds, not with just a therapeutic mindset, because this is an unmet medical need. There is no current therapeutic for this cancer. But as he was screening it, not only did he find potentially new compounds that could be, that he could think about driving towards the clinic, but he learned a lot about the actual mechanisms because of how well the library is annotated. He had clusters of compounds that were hitting the same target or hitting the same pathway, and it unlocked a new path for him to go down, so that maybe if he can't target this directly on pathway, he could now go after a different pathway to target to see if it would affect these cancers. One of the biggest projects that has used this and is actually also a resource for the rare disease community is this Jump Cell Painting Consortium. So in this cell painting is a imaging assay that was developed at the Broad. And in this consortium, they had a bunch of different partners, industry, academic, government, and they took 116,000 small molecules and they screened it through this assay in U2OS cells. Now, U2OS cells are a bone osteosarcoma cell line. They're not necessarily the best model for every disease, but they are a starting model, and the reason they're chosen is because this works really well in this imaging assay. For 116,000 compounds, 4,800 of them came from our repurposing library. And the idea is that you treat the cells with these compounds, you cell paint them like this. So you have these very, very pretty images. And then you can actually extract features from these images using their cell profiler. And for every compound treatment, you can basically create a novel profile of what is happening phenotypically in these cells. So some of the repurposing compounds are in there. You see the control on the left and then the different treatments. Some are very obvious. They elicit a very different phenotype. So you can imagine the feature profile is very obvious. But then some of them, like this, are not the most obvious by eye, but using computer programming, you can extract that there are differences in those features. So right now, they did compound screening, they did overexpression, and they did CRISPRs of different genes. And so they're analyzing and compiling all of that data. Right now, you can do comparisons within each perturbation, so within ORFs, within CRISPRs, within compounds, and they're working on the idea of being able to cross compare the perturbagens at this point. So the reason I bring this up is because you can imagine that we have a multitude of rare disease foundations that come in to ask, can I use your repurposing library to fix my disease? And so the hardest part about this is having the right platform to screen the repurposing library. And we can't just throw 5,000 compounds into a human being. That's pretty frowned upon by the FDA. So we need something to screen, whether it's a cell-based assay, whether it's biochemical-based assays. And so many of these foundations, they've learned a lot in their time of being a foundation. And so many of them have actually learn from each other that the first thing you need is probably a cell, a set of cells, whether they're fibroblasts or iPSC. So they're at least coming to us with that, but they don't necessarily have an assay in which to screen. What does your disease cell look like versus a normal cell, and how do we fix that using the repurposing library? So we will work with a lot of these foundations to help build that assay. So you can imagine then that if you don't have a phenotype, if you don't know what you're looking for, you could use the cell painting assay. So an example here is the ZTCK syndrome that is caused by a low expression of the SON gene. So it just so happened that the SON gene was used in the cell painting consortium. And if you knock out SON to mimic what the disease might potentially look like, you get a phenotype. This is a pretty obvious phenotype. You can definitely see there are changes between that and the control cell line. So right now, because you can only do within perturbogen comparisons, all you can ask is, are there other genes that were knocked out that have a similar phenotype? And the answer is yes. So this doesn't necessarily mean anything to me right now, but it's possible that if you dig into the literature, you may see connections between the gene of interest and other genes that are playing, that are giving a similar phenotype. But the cool thing is you can ask the question, what are genes that are knocked out that give the anti-phenotype to what I'm looking at? And there are genes that give the anti-phenotype. So interestingly, these were all TAF genes. And if you looked at the whole list, almost every member of the TAF family came up. So the question is, is there a mechanism in the TAF pathway that if you perturb that, you might be able to get the anti-phenotype to your gene of interest? So again, as I said, they're currently working within perturbations, but then you could imagine that once they do the repurposing library profiles and they start being able to do comparisons, could you look for a repurposed compound that elicits the anti-phenotype or a similar phenotype? And could you learn something about the biological pathways that might be involved in your disease that you wouldn't have necessarily known without using this sort of unbiased approach? And then finally, one of the more interesting projects we're working on now is actually a pediatric cancer drug screening project. Pediatric cancers as a whole are considered a rare disease, and obviously any subset of pediatric cancers is a rare over rare. We are working with Greg Way at the University of Colorado on a 4-year funded project by Alex's Lemonade Stand, which is a pediatric cancer foundation. And the idea here was, could we amalgamate a bunch of pediatric cancer cell lines and map the morphology of these pediatric tumor cells? What do they look like at baseline level? If you got a bunch of rhabdosarcomas together, do they all look the same morphologically, or do they look different from each other? And are there underlying genetics that change those? So we are in the process of aim 1, which is to map the landscape of about 50 pediatric solid tumor cell lines using the cell painting process. And the idea here is once we map that morphology, we'll pick a subset of those cell lines and run the repurposing screen library against that in a screen. The goal here is not necessarily to find compounds that just kill the cells, because that's what most cancer researchers wants to do. But pediatric cancers are a little bit different because of the developmental genes and developmental pathways that are involved in pediatric cancers. So rather than looking for compounds that just straight up kill, can we look for compounds that shift the morphology towards a potentially healthier phenotype? So we're starting this project as a screener. We work in mini formats. We're in 384 well formats. We can do this assay in 384 well format, where we have different densities of the cells, different cell lines on a plate, and we can start looking at what the baseline morphology is. Morphology is affected by density. So these are sort of the early experiments that we're doing here. All right, in my last few minutes, I just want to talk quickly about accessibility. We have this, it's available for academics and nonprofits to come use. But there's obviously a cost to screening. So if you are lucky enough to have the reagents that you need and the funding that you need, you can access this library in 2 ways from us. The first is we can ship this library out as assay-ready plates to your institution, and you can run the screen yourself in your lab. We can do this in 96 or 384 well format. You would consult with us first, obviously, to understand what the ideal format and concentrations are for your assay of choice. If you don't have the infrastructure in your institution to be able to run that screen with us, you can work with my group. We're the collaborative screening group, and you can come talk to us about how to develop your assay, and we can take it from assay development all the way to screening and to hit validation for you. But the other things that we think about too are, just because we have this resource, not everybody actually has the assays or the tools or the reagents or the funding to be able to use the resource. Screening costs money. It's just a fact of life. So we are thinking strongly now about how to bring down either the cost of screening, whether that's creating an informer subset. Can we go from 5,000 molecules to maybe less than 1,000 and give people a first taste of how to use the repurposing library? Can we coordinate multiple rare diseases and pool the effort together to run multiple rare diseases side by side to bring down the cost of that screening for each individual rare disease? Pooled compound screening, all of those things are going through our minds right now. can we help you get the assays, the reagents, and the tools that you need? If you don't have cell lines, can you come work with us and members of our network to help you build, iPSCs, fibroblasts, anything like that? Can we partner with some of the other platforms at the Broad to help get some information, whether that's RNA-seq or proteomics? And obviously data sharing, this is a big part. We are trying to build infrastructure so we can share with other people what data is being generated out of their repurposing screens. And finally, just one plug for my department. We're CDOT, we're the Center for the Development of Therapeutics. We're an integrated drug discovery capability platform. We have multiple capabilities. I strongly encourage you to come talk to us. As somebody asked, where do you start? You start with an email where I'm on the receiving end, and I will promise that I will answer, and you bounce your ideas off of us. We can help point you in the right direction. And with that, I will just close by thanking my team, the automation team, the compound management team, our professional wranglers, their project managers, and everybody in CDOT. And there are 2 email addresses down there, cdotcollaborate@broadinstitute.org and repurposing@broadinstitute.org. Feel free to reach out to either one if you have any questions or want to chit chat. And I'm on the receiving end of both of those emails, so I promise I will respond. Thank you.